Uh-huh. Uh-huh. Hoo-hoo. Look at this. Sure. Do you have anything to analyze in your mind? Hmm? Single cell. There's like just RNA sequencing, right? So when we request some data set to eBiogen to get GAC number, data set did we request? There's some metric that's size is not that big, a few hundreds of mega- that's smaller. Data is this is the process data. Yeah, so process data is around few hundred megabytes like that, right? So we are going to upload that one in GSC. Bigger one. Bigger one as well? Mm. Not processed one? Make raw. Uh-huh. Right, we upload it. Mm, got you. So let's start. So you guys already installed this RStudio. There's a couple of things you have to download and install, but all of them. So the thing I want to share with you in today is that if you want to analyze something, sequencing, any sequencing, microRNA sequencing, single RNA sequencing, just bulk RNA sequencing, just ask, okay, I want to do in English or Korean, I want to analyze scRNA sequencing using our studio. What kind of program we have to use and how we can check whether those program is already. You can ask using your AI platform, any Gemini, ChatGPT, or whatever, and then they will kindly, yeah, pay you how you check and how you analyze. So here, just maybe 2 weeks ago, I just randomly picked up the idea. Maybe this AI can guide me how to analyze all of the sequencing data. So I asked like this. So and then they told me I have to install RStudio. This is free, and then we can get a process data in GSCE, which is uploaded in NCBI, and then I can easily access and I can download and analyze it in my way. So, so studio and these packages, all of them, and keep up something like this. So I check these things is already installed. So you just copy this code and then put in your R studio, and then here they check all of the needed contents are already installed well. So this is Korean, but maybe in English you can check. They're all set up. So if you have any like error, something like this, and then you can copy this error and then put here, and then they'll give you some answer. And then you have to correct your code. So, and then today, after installing all of this, I wanted, okay, I want to analyze this system. Yeah, let's see. Where's that? No. Today, for the first time I analyze this data is just, sorry, what's that? Maybe this one. I want to analyze macrophage M0, M1, with M1 with alpha interferon gamma, M2 is then, sorry, what happened? Okay, let's download and analyze just one data. And then they search. So there are two ways to analyze, two ways to download. You can directly access Geo using this code. And then when you copy and paste this code, this is already uploaded in Geo. So anytime when you make some sequencing data for publication, you have to upload the sequencing data in this NCBI, and then you have to get some Geo number. Now it is necessary. So here they already uploaded here. So actually you have to download this one by one. This is not that big, or like few hundred of byte. Here just this is like some is few megabyte, some is few kilobyte. But this, but when you look at this, you can download from the R. So maybe I just follow this. Install this, just copy, and then copy and paste here. And then they automatically access to Geo and then download it. Let's see how. So you just pick up any sequencing data and then follow me right now. For me, I'm downloading this one, GSE 161125. 161125. Okay. Here for downloading, they are installing BioC Manager, this program. And they are downloading and then installing right now. No update. And okay. And then data down here. Oh, bio I can paste here. Now they are downloading this one. Okay. Just see this is matrix. Now they all downloaded. Okay, I finish. And here they show me what is the code for data analyzing. Paste. There is something. Okay. There is some error. Okay. So let's see. If we have certain trouble, maybe there's error here. Copy and paste here. And then they analyze what is the error of this thinking. Oh, okay. They are showing some error. So let's see. So I copy and paste here. Mm. Library, copy, set. Now another one. So when you see this red stop sign, which means they are running right now, this stop sign changed to green something, which means you are ready to do something. Now they are downloading here. So let me manually download it. One. For just in case, download the OR file. M0. And then this O. One, two, three. One, two, three. Last one. One, two, three. Okay. Oh, they were downloaded here. The blah blah blah. They already downloaded. So I copy this and then paste here. And then they check, okay, you are downloading well. Oh, downloading well. So you have to unzip this file. So let's copy, paste. They unzip and then they show a certain list of file. Copy paste here. Mhm. Okay, we have M0, M1, M2, M1, M2, 4 with 10x single cell sequencing file. So nowadays we have to make some file, something. Okay. Now they're analyzing. So copy this one, paste here. Okay, this is some error. Okay. And then copy paste error, copy, paste here. They are showing something. Okay. And then copy, paste. M0 count, M1 count, M2 count, and M1 and M2 count. Let's see. Okay, M0, M1. I have a question. Maybe they have four. Why they are making three? M1, M2. We have this group also, co-stimulating. Fix that. I know this data set have four groups. Then M1, M2 group. They are combining all. Okay, copy. Now they are loading this one, M1, M2. This cell number is like this. They're combining all. Then now they are doing QC, mitochondria. Then here, they automatically make this one, right? This like feature of RNA ink count and percentage. And then if you want to download this, okay, you can download from export, save image, or here you can ask, I want to so I want to download a code. Okay. Then they said desktop, this is downloaded. Okay, copy, paste. When you look at this here, this is PNG file. Here, this is file you make it. Why? Okay, right. And then copy this one, paste here. Let's process, proceed, analysis, analyze, analyze. Okay. They describe what is the meaning of this one, and then generally good. So they are making some filtering outlier, filter and normalization. So this is their features. If you want to change something, and then you can change it, you ask. But I just follow this. They're making filtering, just this outlier bottom up, they remove it. So they are normalizing this layer M0, M1, M2, M1 plus M2. So meanwhile, so I want to know this, what is the meaning of this analyze data? So just I copy paste all of this and then put here. This is, sorry, data set I am analyzing. Explain to me. What's this? So now you are educating your AI. So this is a data set. Now we are analyzing. So maybe you can read all the things and then you can catch the idea, but I want to simply get it. So this is from Nature Communication paper from Yale, bone marrow. And then they just media only, alpha interferon gamma, 4 and M1, M2 all together. So sorry about this Korean, but they said, mm-hmm, okay. They said M1, M1, M2, they're opposite. So when you treat LPS and interleukin together, they are okay. Maybe explain in English. They said they assume that when they treat M1, M2 together, maybe some M1, M2 cell, they are macrophage between M1 and M2. But here they mention the macrophage individually M1 and M2 and then mix them together. So this is their thing. They have already chat with your AI. Sometimes they show you relevance to your research, something like this. Okay, and then now we are finishing this. So here they automatically doing PCA. PCA means how individually you can. So just you copy this one. Maybe you didn't know what's meaning. Well, I know it. So basically, 1, 2, 3, 4, like this, and then you can do PC as like 4, 1, 2, 3, 4, or sometimes here you can just gap here, so 10. So when you make higher PC, which means that you are making higher cluster. So just let's check by this one. What is your recommendation? Like 10 or 5. Oh, they recommend 1 to 15. 110 capturing areas, 115 macrophage, 15 is optimal. So recommend 115. How? 15 is around here. So you can see this drop. Okay. Okay, so I'm gonna follow this. Copy and then paste here. You guys have followed me? Serious? Okay. 동현? 유정이 잘 되고 있어? 따라해봐. 어. You have to follow it. 어. Just click. If you have some error, copy and then ask. Here, they automatically show this. Also, they are downloading here. So let's see. Okay, this is depending on your cluster. Okay, this is depending on your M0, M1, and M2, something like this. Hmm. And then you copy this to why they are far away. So I have a question. Why this M1, M2 is far away from this? Because I assume that they are all clustering. So this is not that much good. So I ask, why M1, M2 is far away from others? Any correction? Yes? Okay. Maybe I already have many experience to look at the single RNA sequencing, so that's why I already know that this is a little bit weird, which are far away each other. So I asked question, so they said batch effect, not biological. So we are doing harmony for making, for decreasing, for making. So just copy this one and paste here. Oh, it's so I copy error here. And they are thinking, oh, but they show another correction. Okay, copy, paste, copy, Version. Harmony version is different. So we have to install and again Harmony. Yes. Now they are installing this Harmony package. So are it done? Oh, it's done. Mhm. So installation is finished. So again, data loading. Maybe I have to make another session, probably. Session, new session, or let me start from this. Copy paste here. That's it. They are already again this M0, M1, M2, M1 plus M2. Let's see. So like that, if you have any data set want to analyze, like this single cell sequencing, bulk RNA sequencing, AT sequencing, and then cells, but cancer cell survival data, or BGM, spatial transcriptomic, any analysis, just ask your AI and then follow their direction. And then you can automatically analyze it. And then if you want to follow certain paper in figure, copy the figure, paste here. I want to make this figure. And then they are going to also direct you how you analyze it using the code. So that's the whole thing. This is one direction how they are doing. So other thing just copy and paste, error, copy, and then correction, also copy and paste. So after making this UMap, maybe I'm gonna finish. And then what is the other one? After making UMap, you have to make what is the meaning of this cluster, right? And then you can ask, they automatically making gene analysis, and then they fit this cell cluster is what's the meaning of this cluster. Also, they are automatically analyzing. You don't need to like analyze all of them manually. Session is time post. Okay. Maybe even though you are making this green color and then this is not appear, just escape. And then let's see. Mhm. Okay. Copy. Maybe I didn't copy all of the thing, and now they are making combine, and that's filtering again, normalizing. So maybe they're going to show us one more time of PC, how many cluster we are going to do. Now they are making harmony to merge them. So depending on your computer memory and then your what's that quality of your computer. If you have fast computer, they analyze very fast. If your computer is slow, they make it slow. So in iTrend, we have analyzed computer. Now back what is that inside of the and behind the Ali and Mosen's sheet, you can use the computer for analyzing. Mm. Now they are ready for showing something. Okay, now they're merging somehow, right? But still, I'm not that satisfied. They are also depart each other. M0, M1, M1, M2. So let's see. This is good. Let's see. This previous one. The, compared to the previous one, this is somehow better, right? Okay. A little doubt about this. I'll ask them. This is their best. Copy this one. Capture. Yes, after harmony. Harmony thing. Mm-hmm. Mm. Okay. They said this is from biological meaning. Yeah. M1, M2 is a little bit different from other M0, M1 only. They are apart each other. But this is way better than previous this one before harmony. This is two different, right? Two different things. But this is somehow close each other. So they said this is good. And then if you have certain doubt about something you're analyzing, you can put your the paper PDF file here, and then based on that they will guide you how you analyze it, and then you can exactly follow the same thing. Okay, now loading. So mhm, library ggplot2. Okay, now they're finished. Ah, was downloading. And then now marker gene imaging. Let's see. They're gonna check this few marker M1 and M2. Okay, I just follow. Now the time for checking what is the meaning of this cluster. Mm, they said maybe all upper one is M1, below one is M2. So M1 is around here, M2 is like this. Just copy this one. And then you're making one PPT. And then all analyzing you can put in PPT to make the story. Here, maybe. Okay, this, your UMap. And then second one is feature, M1 and M2. So you look at this is good or bad. M1 is here, right? This green color. M1, M2 is surrounding here, M only here, M0. So this M1 marker should appear on the right side, right? M1 marker on the right side is good. M2 marker is relatively left side, or this is good. So I think this is good. Okay. And then I want to get, I want to know the meaning. One. Now we have how many clusters we have? We have 9 clusters, 10 clusters from 0 to 9. Okay. Meaning clustering meaning of 0 to 9. Let's analyze. Seems they are showing this. Showing this is to confirm you are doing this U mapping, good or bad. Yeah, for knowing that, they just show as a confirmation M1, M2, they are well localized. Yes, and then we want to know specific what's the meaning of this. So I just asked them. It seems good for U map. Then they guide you how to do that. Mm. They describe M1 marker on the right side, M2 marker is left side, interleukin 4, receptor alpha, M0, M2. This is exactly the same with your paper. So now, to know cluster meaning, let's do DG, differentiate expressed gene. So they found each cluster marker, and then from the top marker, they are analyzing what's the meaning of this cluster and heat map. But I'm not sure 5 markers enough. So based on like 20 top markers, let's analyze. How can I do this? Because I already make analyze based on top 5 gene, but sometimes top 5 is not enough. So just if you increase the number of gene, they are more accurately analyzing. This is based on your gut. So I will change the code here. Okay, copy, paste, some error. Okay, error here. Mm-hmm. So they are correct with their code. Final marker take 2 to 5 minutes. Let's see. Error again. Copy. Mm-hmm. Okay. Click. Oh, now they're making. Mhm. They're saying we are performing certain thing. Let's see. But the good thing is that once you analyze some data and then you already download and install everything, so next one you can easily analyze it because now this is the first time to analyze it in this notebook. So that's why many things we have to install. So there's some error. But once you finish one whole data set, the next one is very easy. Mm. Now they are calculating top 20 from cluster 0 cluster 1. And then they're going to show us like gene set. And then you can copy paste in this gene to your AI. The AI analyze, oh, maybe this is your cluster. And then next time, how you can do, you can also change this one. Okay, based on you change this code to your identify cluster name. And then also like that. Mm. So maybe after 1 hour later, maybe I will analyze all of them. So this is the one how you guys can do analyze based on AI. So in my opinion, I just only use the Claude for analyzing because I heard that Claude is best for giving the code, but ChatGPT or is it Gemini, I don't think you can do anything. Mm. So any question? So you can see how they're analyzing, or you can leave after giving me any question. 동현.
노트북
음,
깔려있는
음,
튜토리얼에서
음,
진행을 한번 했잖아요.
음,
우선 이제
음,
각처에서 분석을 한번 끝내면
다음에 어떻게 하냐?
다음 그 뭐지? 다운로드 이게 다운로드를
것들이.
라이브러리도 많고
음,
그게 다 툴도 다 설치를 해야 되잖아요.
음, 음,
그 설치를 하고 나서
음,
다음 분석을 처음 다운을 받아야 돼서
음.
다른 세션에서 풀면
아, 그럼 어떻게 하냐면은 물어봐 여기서. So yeah, this is very good question. So you analyzing or almost analyzing, that you want to remove, you want to exit, but if you save all of them, the content like around like few gigabytes, very big. So in that case, here, okay, I want to stop here, but so what is the essential essential thing to save? Let me know based on code. And then maybe they're making some RDS file. Oh, they just show like essential file to download it and then just download. And then also you can ask them what is the opening code. And then based on your downloading, they are making an opening code. So you can easily assess for next time. 그렇게 하면 됩니다. Maybe, but it's very important. So maybe time you are analyzing like few hours, and then you don't need to take another few hours for analyzing again. So or some CSV file for knowing gene set or biological process or data, and then next time you automatically analyze it. Here, okay, they are making all things. So, other question? Mm. All set? So next Tuesday, I want you to analyze one single RNA sequencing data by yourself. Okay, any data is okay. Maybe based on your like your research field. Maybe in case of who's that Yunjin, you are into what's that nerve regeneration, right? And then you can look at what you can ask here. What is the uh, nerve tissue signal RNA sequencing data? And then there's something based on that paper, or just based on that think of okay, this GSA data set. Maybe for you are analyze, you can use this data set because you want to generate the right? And then exosome they have certain ingredients, but for that you have to know that what is the what is the or how they are doing at the moment. When nerve, maybe peripheral nerve, you're targeting peripheral nerve or trauma brain injury, right? Trauma brain injury, what's going on there based on single cell sequencing. And then maybe you can enlarge your knowledge like that. Based on your interest, you can pick up one and then analyze and make it, okay? And then refer to that paper, publish. You try to follow the same thing and then exactly follow to Tuesday. And then upload in our group channel. And then, and then meanwhile, maybe if you have such ask your AI, most of the trouble will be okay. Sometimes I feel like there's still the problem that they're far away. And then maybe 4 or 5 times I try copy and paste, but it didn't work. And then I ask them, is there any way, not this direction, could you think another way to simply fix this problem? And then they, oh, maybe I have this solution. And then they just show us another direction. Mm. So believe your gut and then just ask AI that they will give you a code. Oh, they already make this marker. Save. Okay. Uh, let's finish around here. They save it using IDS file. Yeah. IDS file is important. Mm. So any question? Mm. Mhm, 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 mhm. Right, right. Let's see. Mm. Okay. So any question? Okay. Till Tuesday. Okay. Make your own data set. Mm. Maybe if you don't know what kind of data set you are going to analyze, ask AI. My interest field is around here. So what is the best single cell analysis data set? Yeah, from my interest, and then they're gonna suggest something. Analyze that. So maybe you can assess two things. One is like GSC from NCBI, which is from United States. They are most of them they are opening. You can download. Another was like from some what is that? Some GSA. This is from China, but China thing they need some special request. So I request many times, but they didn't allow me to access it. So based on that, maybe some European data set. Most of people they are uploading your file in NCBI, so they can free download it. But other China thing is not open 100%. Right. Now they are making this. Yeah. Good thing is that they are making this cluster very well. Condition proportion per cluster. M0, M1. Interesting. Oh, they're already making it. Mm. Here they make heat map top 20 here. Maybe from here you can see. Back. Give you some time. Maybe you one time you see this kind of heat map, you can see this very little like gene set here, right? Is it here? So this top 20 and the one, this cluster 1, 0, 1, blah blah 9. They are making this cluster and then they are showing this heat map. This yellow color is high expression, violet color is low expression. So maybe they're extremely distinguished from this cluster. This is good, or somehow this and then 0 and 3 they are combined here, but this is different. So okay, it's quite relatively fine. And then they're gonna show us like this some data set, maybe top 20 data set here or markers. Mm. And then you can assess this clustering is good or bad. If you have some doubt about this, just copy these images. Gosh, where is that? But I did it out. M0M, okay, it's fine. Mm. So if you have certain doubt about this, and then you can put your image here, analyze imaging to confirm this is good. And then based on top 20 gene, analyze the meaning of cluster, each cluster. Even though I'm writing in English, you can write in Korean, any language you prefer. And then mm, they're analyzing on this is make something from top 20. They're making biological meaning of cluster. And then now we are extracting top 20 cluster here. Okay, this is the top 20. Copy and paste here. Based on that, here, this AI set, they analyzing what's the meaning of this cluster. Without AI, maybe you guys have to analyze one by one from the top 20 gene. Okay, what is the meaning of this? And then you have to study based on the paper, and then, oh, you're not that expert in that area. It's not easy to see what is the meaning of this gene. And then it takes a long time. But now you have AI. Ask them. Within 1 minute, they're going to show you the result. Even though it's your first time to visit that field, like nerve tissue, you are first one, just ask, and then they're going to give you very good suggestion. Here, they said key marker identify resting macrophage, stimulated one, inflammatory, proliferating, metabolic active, something like this. If you don't like this one, and then you can, okay, what's the difference between interferon gamma, interferon stimulated macrophage and inflammatory macrophage? You can ask them, and then maybe this 10 cluster is not good, and then you can go back. Okay, I have to make a little shorter cluster, like 4 or 6. So based on that, you can just ask them, and then try to tune your this. Mm. Here the class biology M1, maybe interferon gamma and FPH is really different, even though you are treating these two different cytokines to make them M1 macrophage. But depending on which kind of cytokine you're treating, so that's why they are making two different clusters. The proliferating, maybe based on the marker, and M2 straight dying. When macrophage is experiencing some LPS or bacteria, cytokine that can be dead. So this dying cell also. Rare migrating subset. Maybe it's migrating. So suggest that they are making this cluster. Okay. Based on that, okay, and then a copy. Maybe this is the last one. I can read you. Then they are making new UMAP after annotating this like this. This is very good. Okay, right? Mm. Now you're good. Okay, so I want you to follow after finding data set. I want to follow that same data set in the paper, or you can ask directly what's going on there, and then you already have your plan. Maybe why you are choosing that data set, and then based on that, based on your ongoing study and then your data set, you can try to link. And next, if you analyze this data set, you can put this data set in your paper supplement list. Maybe what is the meaning of your our study? Well, why you are starting your study? Maybe based on in this case, when I look at the trauma brain injury tissue level and sequencing data, they said some maybe neuron cell is dying. This is very relevant. But now neuron cells dying. You're just analyzing that single cell and sequence data set in public, and then based on that, you really die. And then they need some specific nutrient, right? Helping signal. That's why we are making this exosome to treat it because this exosome is already have some anti-RS or something like that. Very starting point. Or the last figure, you are making certain thing, and then how you will. What is the meaning of this in clinically? You can't do all the clinic data things, clinic study by yourself. We have limited time. Because public data set they have. So when I analyzing in real trauma situations, neuron cells die. So we already confirm our exosome have certain microRNA related to antivirus. So that is why they could help in clinical way, right? So like that, mm, you analyze the data set in this time, you guys can put this data set in paper, and it's in supplementary. Also bacteria as well. Bacteria, you can find bacteria sequencing data, single cell sequencing or anything. Okay, thank you. See you next Tuesday. Mm. Next Tuesday, bring